Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. I hope you will make use of it. Thank you very much. In the last class, we have discussed the history and epidemiology related to hepatitis B virus. So today we shall continue with the causes of hepatitis B virus. The infection can be diagnosed about 30 to 60 days after the exposure. Once you get exposed to it or transmitted disease, that means virus enter your body, usually after 30 to 60 days after exposure, we come to know that, yes, that means one symptom will be there, first symptom will be there. The diagnosis is usually confirmed by testing the blood for the parts of the virus and for the antibodies against the virus. Now, one of the best part of this particular hepatitis B virus is it produces so many antigens. It has got so many antigens: surface antigen, core antigen, E antigen, polymers, etc. So now all the antibodies in the body will going to produce against all these antigens. By estimating that antibodies, we can say what is the state of the disease. So it is one virus with and that means this is one of the 5 main hepatitis viruses. We know that hepatitis A, B, C, D, E, etc. is there. Among all them, the most dangerous is hepatitis B and also D to some extent because it is also DNA virus. The infection has been prevented by vaccination since 1982. We know that vaccination after 1982 significantly reduced the incidences. The vaccination is recommended by the World Health Organization in the first day of the life if possible, because we will go for 0 dose on the first day itself. Okay, hepatitis B vaccination schedule, if you see, 0 dose will be given on the first day itself. Now 2 or 3 more doses are also required. That we'll discuss in the vaccination part in detail. The booster doses are required to give the full effect. The vaccine works about 90% of the time. Almost all a safe vaccine and most efficient vaccine. About 180 countries give the vaccine as a part. Now, it is also recommended that all the blood be tested for the hepatitis B before transfusion, and that condoms and the condoms to be used to prevent the infection. Now, this is very important thing. Nowadays, any surgeries before blood transfusion or anything, it is almost all mandatory to test for the hepatitis B. Okay, and to prevent sexual contact, transmission through the sexual contact, condoms are compulsorily used. During an initial infection, the care is based on the symptoms that a person has. Now, in those who develop the chronic disease, antiviral medications such as tenofovir and interferon may be useful. However, these drugs are very, very expensive. Details we will see later. Now, just I am giving the outlook. Liver transplantation at some time used for the cirrhosis. Okay, about a third of the world population, 1/3 of the world population has been infected at one point of their life. That means a big number. Repeatedly, I am telling 1/3 of the population have got the risk of getting infected by the hepatitis B at one point of time. At least 395 million people, 5% of the world population had a chronic hepatitis B virus infection by 2017, and this is almost in the last class also we have repeated. Now, talking about the hepatitis B, now hepatitis B virus is a member of Hepatonavirus family. The virus particle or virion consists of an outer lipid envelope and an icosahedral nucleocapsid core composed of the core protein. So this has got an outer cover which is covered by the lipids, and inner cover is there, inner core area is there, and it's composed of a core protein. So outer cover, that is lipid, so that becomes the surface. So it has got an antigen which is called a surface antigen. In the core there is few more antigens are there which is called as core antigen. The virions are about 30 to 42 nanometer in diameter. The nucleocapsid encloses the viral DNA and the DNA polymerase that has the reverse transcriptase activity. Now, in the core area, the nucleocapsid, that means the DNA where it is present, it encloses the viral DNA. It contains the DNA. It's very unusual to have DNA in a virus. Only few viruses will have a DNA. Otherwise, all the viruses, fortunately, they are RNA virus. So therefore, they're self-limiting character. Now here it is DNA virus. Okay, now in that particular core area we will have the DNA, and there is also a protein called as DNA polymerase. And the specialty, the game changer is this DNA polymerase, because this DNA polymerase has got reverse transcriptase activity. Now this is responsible for duplication of the virus. Otherwise the virus will going to die after once get explode, but it will not happen here because it's a DNA virus, number one, and it also contains a DNA polymerase which has got reverse transcriptase activity. Even HIV virus also has got a reverse transcriptase activity, but fortunately it's not a DNA virus, but it just contains a reverse transcriptase enzyme, and that is repeatedly doing the replication. Anyway, we'll discuss later. Now, the outer envelope contains embedded proteins that are involved in the viral binding and the host cell and entry inside the cell. Now, this outer envelope contains lipid layer and all. It contains some proteins. Okay, so what is the function of these proteins which is present in this outer cover? So they helps in the viral binding, and it also helps to enter inside the host cell. For example, hepatocytes, host cell, and entry inside the cell. So how to enter inside the hepatocyte? So that will be guided by the proteins which is present in the outermost layer. The virus is one of the smallest enveloped animal viruses. This virus is the smallest enveloped animal virus. The 42 nanometer virions which are capable of infecting the liver cells, which is called as hepatocytes, we know that. These viral particles are also referred as DAN particles, D-A-N-E, DNA particle. Okay, these viral particles, yes. Meanwhile, in the blood, apart from the DAN particles of infected person, sometimes we also find filamentous and spherical which are lacking in the core area. This forms the outer part of the virus and produces the antigens called as surface antigens, and it is produced in excess during the life cycle of the virus. Now, what happens is the outer part of the virus, that is the inner layer of the outer part of the virus, it also produces an antigen which is called a surface antigen, and this is produced when it is produced in excess, when the virus is completing its life cycle. In that time, these antigens are produced in excess. See, this is that whole hepatitis B virus. This is the outermost layer. And it contains the M, S, L. So these are all the surface antigen, HBS surface antigens which are produced. Okay, and this is the core area. Okay, this greenish area. So this is a core area. So core area produces its own antigens. That's called core antigen. Okay, and this is the dangerous polymerase, DNA polymerase. So here, this is a portion where the DNA is there. Hepatitis B virus, DNA is there. In that DNA, we will have one protein which is called as polymerase. Okay, this is called as DNA polymerase, and this is a game changer, and this is responsible for all the activities. It has got a reverse transcriptase activity, and therefore this will going to do all the damage. Now, transmission of the hepatitis B virus results from exposure to the infectious blood or the fluids containing the blood. It is 50 to 100 times more infectious than the human immunodeficiency virus, HIV virus. The possible form of the transmission include sexual contact, blood transfusion, and transfusion with their human blood products. Reuse of the contaminated needles and the syringes repeatedly, I'm telling, and the vertical transmission from the mother to child during the childbirth. Now, without intervention, a mother who is positive for HBsAg has 20% risk of passing the infection to her offspring at the time of the birth. The risk is as high as 90% if the mother is also positive for HBeAg antigen. Okay, this is a type of antigen, core antigen. So HBV can be transmitted between the family members within the households, possibly by contact of a non-intact skin or mucous membrane with secretions or saliva containing HBV. Now, however, at least 30% of the reported hepatitis B among the adults cannot be associated with the identifiable risk factor. Breastfeeding after breastfeeding after proper immunoprophylaxis does not appear to contribute to the mother-to-child transmission. Very important. That means if the mother is given proper immunoprophylaxis, then breastfeeding can be carried out safely. The virus may be detected within 30 to 60 days after the infection and can persist and develop into chronic hepatitis B. The incubation period of the hepatitis B virus is about 75 days on average, but can vary from 30 to 180 days. The genome of the HBV has got made circular DNA. See this in this the DNA. This is circular DNA. This is not network like. It is circular DNA, and but it is unusual because the DNA has not fully double stranded, isn't it? See, the DNA is not double stranded, only single stranded, but it is circular nature. The game changer is polymerase. So this will do all the remaining work. Okay. Now in the next class we shall see details of structure of the virus. Thank you very much.